Hi micro folks, we are doing chapter 17 metabolic tests part 2 results. In this video we'll do IMViC results, then we'll do another video for the TSI and a last one on catalase. So folks, again, it's really helpful if you can read the lab manual chapter 17. The IMViC tests is a series of 4 tests: the indol test, methyl red test, Proskauer's test, and citrate test, that lets us distinguish E. coli, which is an indicator of fecal contamination of food or water, from its close cousin, Enterobacter Orogeny. Okay, so if we find high levels of E. coli in food or drinking water, we're really worried about fecal contamination. And we know if feces are present, any intestinal pathogen could be present. So viruses like norovirus, hepatitis A virus, poliovirus, bacteria like Vibrio cholera, Salmonella, Shigella, E. coli 157H7, protozoal pathogens like the cysts of Giardia or Entamoeba histolytica. We could have helminth eggs like Ascaris lumbricoides or Taniosolium. So if we find lots of E. coli in food or drinking water, we want to immediately put out, a public health alert: stop drinking the water, stop eating the food, because folks could become sick with a number of different pathogens. Now the challenge is that E. coli and its cousin Enterobacter Orogeny, they initially look similar, and the reason it's important to distinguish them is because Enterobacter Orogeny can live in the intestinal tract, but it might also be found living in dead or decaying organic matter out in nature. So if we're only finding lots of Enterobacter Orogeny in, say, drinking water, but no E. coli, it suggests it might not be fecal contamination, but maybe just some environmental contamination. So the reason the IMViC tests are so helpful is that E. coli has the exact opposite test results as Enterobacter Orogeny. So just really quickly, you guys, and you'll want to memorize this for the lab exam too. So the IMViC results for E. coli are positive, positive, negative, negative. Positive indol, positive methyl red, negative Proskauer's, negative citrate. And then for Enterobacter Orogeny, it's the opposite. So Enterobacter IMViC is negative, negative, positive, positive. Enterobacter is negative indol, negative methyl red, positive Proskauer's, and positive citrate. So folks, let me just really quickly go through the, using our photo atlas, go through the indol test. So folks, the indol test it's a test for the ability of your microbe to make indol from the amino acid tryptophan. So really what we're trying to detect is, can your microbe make tryptophanase? Does it have a gene to make tryptophanase? And in the old days, we had to use these liquid media, but now, you might recall in the first video on chapter 17, we have these cool little dry slides, over here, that have all the reagents to detect tryptophan. And so we said, what we did is we harvested cells from our plate and then smeared them onto the surface of the dry slide. And if the reagents turn pink, that tells us that indol is present, and that tells us our microbe makes tryptophanase. If we smear our cells onto the indol slide and it doesn't turn pink, that's a negative indol test. No indol was made, and it tells you your microbe doesn't make tryptophanase. So for E. coli, folks, remember, E. coli is indol positive, so we would get a pink result with E. coli. Enterobacter Orogeny was negative, so we get like a yellow result for Enterobacter Orogeny. Okay, folks, now the next test is called the Methyl Red Test. And the Methyl Red Test lets us detect if our microbe uses a specific fermentation pathway called mixed acid fermentation. And as the name suggests, the product of mixed acid fermentation is a number of different acids. So lactic acid, succinic acid, pyruvic acid, and as a result, the pH of the medium is going to be really, really acidic. And the final pH, you guys, if mixed acid fermentation is being used, the pH should be below pH 4.4. So what we're going to do is add this cool pH indicator, methyl red, and methyl red pH indicator is going to turn red below pH 4.4. So folks, when we add our methyl red pH indicator, and you'll recall, folks, that on Friday we had inoculated our E. coli into our methyl red broth. This is a high glucose broth. So if E. coli carries out mixed acid fermentation, when I add my methyl red pH indicator, if the media turns red, then I know that I have a really low pH, and it tells me that my E. coli carried out mixed acid fermentation. So I'm just adding a few drops of the methyl red pH indicator, and I'm going to shake it gently. And folks, see how it is indeed red. Oops, sorry, need a prehensile tail. So this is telling me that my E. coli, because it has this really low pH, the methyl red turns red, so that means the pH is below 4.4. This tells me that my E. coli carries out mixed acid fermentation. So we call it MR positive, methyl red positive, and the interpretation is it's using mixed acid fermentation. Now, in contrast, folks, we're going to find out Enterobacter Orogeny uses a different fermentation pathway, not mixed acid fermentation. So when we add our pH indicator to our Enterobacter Orogeny, we're predicting it shouldn't turn red because it doesn't use mixed acid fermentation. So let's see what happens when we add our methyl red. Okay, 1, 2, 3, 4, 5. Okay, there's that great old Xfinity advertisement. Not sure how we got targeted for that. Okay, so you guys, let's see what happened to our Enterobacter Orogeny and methyl red. And folks, see how it didn't turn red? Okay, so that's MR negative. That tells us Enterobacter Orogeny doesn't use mixed acid fermentation. Okay, all right, so far so good. They read the lab manual while they were incubating. Good. Okay, folks, the next test of the IMViC test is the Vogt-Proskaris test. And we're going to dry lab this. You'll recall, you guys, Friday we inoculated E. coli into VP medium. Again, it's just a high glucose medium. And we also inoculated our Enterobacter Orogeny into VP media. But folks, to run this test, you have to add some pretty toxic chemicals, alpha naphthol and 40% potassium hydroxide. So we're just going to dry lab it. So folks, if we added our alpha naphthol and 40% potassium hydroxide to our E. coli VP broth, there should be no color. And this is telling us that E. coli does not use butylene glycol fermentation, also known as 2,3-butene-diol fermentation. In contrast, folks, if we added our VP reagents to our Enterobacter Orogeny tube, we would see a red color develop within 30 minutes. And this red color is detecting a unique intermediate of butylene glycol fermentation. Its fancy name, the intermediate's name, is acetylmethylcarbinol, but a nickname, shorter name, is called acetoine. So the red is detecting that acetoine. That tells us that Enterobacter is using butylene glycol fermentation, right? So it's VP positive, just as we would predict. One more test, you guys, in the IMViC series of tests, and that's the citrate test. And this is a test to see if your microbe can use citrate as a sole source of carbon and energy. And the pathway to me is really complicated, so we're just going to stick with the simple things. So you guys, the medium that we used to run our citrate test, it's called Simmons citrate agar. It's got this really cool pH indicator in it called bromethymol blue. And if citrate is used as a sole source of carbon and energy, we'll get an alkaline pH, a high pH. And the bromethymol blue is going to turn this beautiful deep blue color, not red, deep blue color, sometimes called Prussian blue. I think of it as Levi jeans blue. And if our microbe can't use citrate as a sole source of carbon and energy, well, the medium will just remain green. So let's take a look, folks, at our little E. coli. And you can see this is after 48 hours of incubation. The medium is still green. So E. coli is citrate negative. Can't use citrate as a sole source of carbon and energy. But then, you guys, this is my favorite one here. Here's our Enterobacter Orogeny. Isn't that beautiful? So the blue color is telling us that Citrobacter Orogeny can use citrate. This is a positive citrate test, right? Okay, so there we go. So again, folks, just in summary, we can distinguish the fecal indicator E. coli from Enterobacter Orogeny by doing the IMViC test. E. coli is positive, positive, negative, negative. Enterobacter Orogeny is negative, negative, positive, positive. And again, you guys, this is really important in detecting fecal contamination of food or drinking water. The next video up, you guys, is going to be on the TSI, our TSI slants.